Okay, so welcome everybody to this week's edition of PaleoPerks. My name is Raymat and I will be hosting the seminar today. So we're really excited to be joined by Ronnakrit Rattanasriampaipong from Texas A&M University in the United States of America. And Ronnakrit will be presenting a talk entitled Untapped Potential of Archaeal Lipids Beyond Ocean Temperature Reconstructions. So if this is your first time at PaleoPerks or you haven't been for a little while, here's the format of today's seminar. So we will have welcome and announcements for about 5 minutes, which is what I'm doing now, followed by Ronnakrit's talk, a moderated Q&A, and an after-talk tea time, which Ronnakrit has very kindly agreed to stay for. So we really encourage you to stay and continue all the conversations about the science in the talk. And don't forget to send your questions via the chat to the questions at PaleoPerks host, who today is Jana. So a little bit of housekeeping. So PaleoPerks values the participation of everyone interested in the paleosciences. Please remember to abide by our code of conduct at today's seminar. If you somehow found yourself here without having signed that, please take a moment to go to our website and have a look and familiarize yourself with this. Please remember to mute yourself for the duration of the talk. You shouldn't be able to unmute yourself, but if you find that you can, please remember not to, so that we can maintain a quiet environment for our speaker. You can ask questions by chatting to the questions at PaleoPerks host or by using the raise hand function as well. So if you raise your hand, we'll unmute you and you can ask a question by voice. Any technical issues should also go to the questions host. We have closed captions built into our Zoom, and you can use the CC button at the bottom of your screen to show or hide them. Please remember to nominate all of your outstanding early career friends and colleagues for our talks. We really like seeing who comes through to our pool. And we also have our weekly feedback form for demographic information, so we know a bit more about who's attending today. So this is anonymous, optional, but very much encouraged, and you'll be able to find this in the chat window very shortly. We also would like you to continue to feed in our participation feedback survey so that we can help to serve you and our community better. The link will be in the chat very shortly, or you can also scan the QR code that's up on the screen just now. So now it's my great pleasure to introduce today's speaker, Ronnakrit Rattanasriampaipong. So Ronnakrit did his bachelor's and his master's at Chulalongkorn University in Thailand. His bachelor's was in geology and his master's was in petroleum geosciences as part of an international program. And he's currently a doctoral candidate at Texas A&M University in the US, and also a Fulbright Thai graduate grantee and Schlanger Ocean Drilling Fellow. And now I'll hand over to Ronnakrit.
Not sure. Is it okay now?
Yeah, it's
all right.
Just loading for. Yeah.
Thank
all
you.
Good.
Thank you for such a great introduction. And I'm Ronnakrit Rattanasriampaipong, but I go by Ronnie for most of you who are not comfortable pronouncing my last name. And it's my honor to be here with the PaleoPerks virtual seminar, and I would like to thank whoever nominated me. I still don't know who nominated me, but I'm glad to be here, and we'll try to share some science today, and we'll learn a little bit about what I am passionate about, what I'm enthusiastic about. So today we will talking about archaeal lipid biomarkers. As you can see from these pictures, it's a membrane spanning lipids that archaea synthesize as their cell membranes. And there are very many interesting applications that people using organic geochemists using these lipid biomarkers to study environmental changes in the Earth past history. But today we will talk a little bit more specific about how we can use these lipid biomarkers to study evolution and ecology of the archaea who synthesize the biomarkers beyond the traditional applications that people normally use them as a sea surface temperature reconstructions. And okay, let's start it. So here is an outline for my talk today. I'll try to go over all of these questions. So we'll start with what are GDGTs and what make them popular, what make GDGTs popular. And then we're going to talk about the results from our recently published paper, how we can use these GDGTs to trace archaeoecology, and what we can learn, or what do we learn from these new insights. So a little bit of a background for GDGTs for those who are not using these lipid biomarkers. So these are glacial-dialkyl glacial tetraethers, and it is one of the most commonly used organic proxies to reconstruct sea surface temperature over the past 200 million years, as they are very well preserved in the marine sediments. So normally paleoclimatologists use these lipid biomarkers to reconstruct sea surface temperature over the past 200 million years. But it's rarely used to study about themselves, about how they lived in the past ocean and stuff like that. So that's why we are interested in looking into how we can use them to infer how the ecology and evolution of these organisms. But before we do that, let's talk about why we are interested in these organisms. So the producers of GDGTs are what we know now is ammonia oxidizing archaea, or AOA. And these organisms are key players in marine nitrogen cycles. We know that they live throughout the water column from shallow oceans to abyssal depth, so down to like 1,000 meters or more. And these archaea are responsible for the first step of nitrification in the marine environments, converting ammonia to nitrite before having other groups of microbes helping converting nitrite to nitrate. So these is a key steps in the marine nitrogen cycles, and that make AOA become a key players in the marine environments. So now let's talk a little bit more about how we use them as a sea surface temperature first, before we use them as an archaeoecology tracers. So these are 6 common structures of the GDGTs that we normally found in the marine environments, in the marine sediments. So if this is your first time looking at GDGTs, it's quite a huge organic molecules. There are 86 carbon atoms in these GDGTs. And archaea is really great in adapting to extreme environments, as we know that they can live in hot spring environments, and they can live in the very high pressure in the very deep oceans. And they are be able to live in such a wide range of environments because they can adjust their membrane structures by adding rings into these lipid membranes. So when there are no cyclopentane rings in the structures, we call GDGT zeros. But when they add more cyclopentane rings, we call them GDGT1 for 1 ring, 2 for 2 rings, and so on and so forth. And for marine ammonia oxidizing archaea, they synthesize 1 special molecules that are a potential biomarkers for the groups, which are these crinochial, which is the GDGTs with 4 cyclopentane rings and 1 additional cyclohexane rings. And whenever we found crinochial in the marine environments, we normally infer that these marine AOAs, the primary producers of the lipid biomarkers. And people observe these temperature-dependent behaviors, like more temperature, higher temperature, more rings in the lipid membranes. And these are what we call homeoviscous adaptation. People found in mesocosm studies, people found a relationship in the culture experiments. But the main application is derived from the relationship between surface marine sediments and the lipid biomarkers that we extracted from the sediments. For an example, here is the liquid chromatograms that we extracted from the surface marine sediments that we're collecting from cold regions. For an example, raw seas, where we have surface temperature closer to freezing temperatures. The main features that we will see on the GDGT distributions are we normally have high abundances of GDGT zeros and relatively lower for the Ronnakrit, very few abundances of these minor GDGTs 1, 2, 3 and Ronnakrit isomers. But if we move our study sites to a much warmer sea surface temperature regions like tropical seas, like Sulu Sea in this case, we can see a significant shift of the distributions of GDGTs from having a much lower GDGT 0 now, significantly increasing these minor GDGTs 1, 2, 3 in Ronnakrit isomer. And with these obvious relationship between sea surface temperatures and the GDGT distribution collecting from surface sediments, Stefan Charlton and others from NIOS groups have proposed the ratios between minor GDGTs as presented here, and we normally call them text 86 ratios. And after the paper came out, people have used the ratios and tried to establish and improve the global calibrations between these marine core top text 86 values with the sea surface temperatures. As you can see on the right is the graphs show the positive correlation between text values from global core tops and sea surface temperatures with color highlighted samples from different regions. But in our talk today, we're not going to talk a lot about sea surface temperature reconstructions. We're going to talk about archaeal ecologies. So before we do that, let me introduce you another ratios which is GDGT2 to GDGT3 ratios. And this is the ratios that introduced back in 2013 by Kyle Taylor and others, which Richard Pancos, who is Kyle's advisor at the time, published a paper talking about how they observed changes in archaeoecology throughout the water columns. But let's think about 2 to 3 ratios for a minute. So as we already learned that GDGTs, archaea add rings to GDGTs when temperature increase. So if we going to have samples from higher sea surface temperature regions, the expected values of the 2 to 3 ratios in these temperature dependent behaviors is going to be lower 2 to 3 ratios. So at higher sea surface temperature, we expected to see lower 2 to 3 ratios, and we think archaea going to produce more 3 than 2. But what we see in the global core top data set is quite the opposite. So this is the exact same global core top data set that people normally use for sea surface temperature calibrations. But when we plot 2 to 3 ratios versus sea surface temperature, it shows the non-thermal behavior. So now we have 2 patterns. We have thermal and non-thermal. And what interesting from the Taylor and others in 2013 paper is that they found that these increase in 2 to 3 ratios rather than controlled by sea surface temperature, it's increasing with water depth of these our samples, our surface marine sediments. So the deeper we go in the oceans, our marine sediments contain much higher 2 to 3 ratios lipid distributions. And that telling us something. It's telling us that we might have some contributions from the archaea that live in the deeper communities. These agree with the archaeal studies that collecting samples from suspended particulate matters in the water columns. So this is pictures from simply modified pictures from paper Villanueva back in 2015, where they study archaeal key genes. For an example, the genes that archaea used to synthesize the GDGTs, and they see a significant shift in the concentrations of the archaeal key genes between shallow water masses and deep water masses, corresponding with the increasing in 2 to 3 ratios. So now we pretty sure that having a higher 2 to 3 ratios meaning higher contributions of the archaeal community that lived in the deeper water masses. So let's have a quick recap of what we just learned. So we learned that we know that text 86 tracked sea surface temperature, and that's what we normally use GDGTs for. We use them as a sea surface temperature proxy. But we also learned that 2-to-3 ratios has a potential to trace shifted in archaeal ecology. But not a lot of studies use these 2-to-3 ratio to trace the archaeal ecology shift in the past, and that's our main goal for our paper. So we wanted to establish a data-driven framework that will allow us to utilize these GDGTs that people measured a lot over the past 20 years to trace ecology and evolution of marine ammonia oxidizing archaea. So in our paper, we do a data compilation from different archives. So we have GDGTs from culture studies. We have GDGTs from hot spring algal mats. We have GDGTs from SPM studies, core top sediments, and ancient marine sediments. And on the right-hand side is just a reminder for everyone that now we're going to look for two main patterns. The one that we call thermal patterns increases in temperature. Right now, we're going to use text 86 instead of sea surface temperature to be able to compare these patterns across different archives because we don't have sea surface temperature for marine ancient sediments. So we're going to look for two patterns, thermal patterns that increase text values, lower 2-to-3 ratios, and non-thermal patterns when the 2-to-3 ratios deviated from the expected trend. All right. So now I'm going to show you the distribution patterns from these different archives. I will group them into three main groups. So we're going to have the modern hot spring systems highlighted in reds. We're going to have modern marine system highlighted in blue. And then we're going to be looking at the ancient marine sediments that highlighted in greens. And let me start with the modern hot spring system. So these first three panels clearly show us that the hot spring GDGTs derived from these hot spring system, either cultures from Pinakiota or cultures from thermophilic ammonia oxidizing archaeas, shows a pretty strong thermal behavior. So increasing text values, decreasing 2-to-3 ratios, which agrees with what we see in the hot spring environmental samples. And these agreements between culture studies and environmental samples confirming us that what we see is something that we think it's a true temperature dependent behaviors. So let's move to the modern marine systems. In modern marine system, we have SPM and core tops. And in our studies, we group the data into shallow water and deep water based on the pycnoclines. So we use pycnocline and group them into shallow and deep. And what we can see here is that on the panel E and F, which are samples, environmental samples from shallow marine environments, comparing to the culture experience of the shallow marine strains, shallow strains of the ammonia oxidizing archaeas, and all of them shows a good agreement of thermal patterns. So cultures show strongly thermodependent behaviors, as well as the shallow SPM and deep core tops. But while we're moving to the deep water environments in these lower two panels, deep SPM and deep core top, we can clearly see that the 2-to-3 ratios deviated from our expected trend. So it's clear that in the modern environments, whenever we go into the deep waters, we have these deep signal that distorted the temperature dependent behavior that we would like to see in our GDGTs data. So to give you a clearer pictures here on the left-hand side, I draw you a schematics of the water columns. And for shallow samples that I have been talking about is the samples that we're collecting from these above pycnoclines regions. So you can see that GDGTs from one ecotype or one clade of marine ammonia oxidizing archaeas shows a pretty strong thermodependent behaviors, while when you have a mix of the lipid biomarkers that synthesize from different ecotypes, you're going to have these like deviated patterns or non-thermal behaviors. So this is what we learn in the modern system. So what we going to see in the ancient is something we'll talking a little bit about. But what we also do in this paper is that we label shallow and deep is quite subjective to our interpretation. So we also confirm whether these different distributions are statistically different. So we use the unsupervised clustering algorithm known as Gaussian mixture model or GMM to confirm that these lipid distributions are different between different water mass by using GMM to cluster the lipid data without informing the water depth. And they're still able to cluster the data into shallow water masses and deep water masses. Agree with what we originally see when we label the data as shallow and deep. So we it's quite clear that these lipid distribution in the shallow water system and deep water system are quite different, and we can distinguish them with the unsupervised clustering tool. So that's good. So we know that it's quite different. And now is what we're going to looking at the ancient marine sediments. So now, over the past 190 million years, people collect samples, extract GDGTs, but I grouped these ancient marine sediments into three main geologic intervals: late Cenozoic, early Cenozoic, and Mesozoic. And you can see a pattern evolve through time, right? So we have these strongly or predominantly thermal behaviors in the older sediments, while we're moving to the younger marine sediments closer to modern system, we have these stronger deviations from the thermal dependent or PTD trend. So let's take a deeper or closer look. So to avoid some biases from our interpretations, we also use GMM crushing for ancient marine sediments or ancient GDGTs. And we have these two clusters, orange and blue. And it's clear that orange reflects thermal dependent, while blue clusters capture the non-thermal dependent behaviors. And when we're going to plot these over time and see how it's changed. Now is the time series of the 2:3 ratios with the GMM cluster results. So on the left-hand side, it's just a reminder, orange reflects thermal dependent groups, while blue reflect non-thermal dependent or a mixed signal. And you can see that we have a very high abundances or high amount of samples that show low 2:3 ratios thermal behaviors during these from early Cretaceous to early Paleogenes, which is a period of time that we know the climate are much warmer than the other intervals. And it's interesting to see that we have a good mixture of these orange and blue data points in the early Cretaceous, while the climate were relatively colder than the late Cretaceous. And we have a strongly increase in the 2:3 ratios values in the ice house climates when we move to these late Paleogene or Neogene. And these observation that we see here kind of inform us that the distribution of the GDGTs or the 2:3 ratios patterns that we observe in the marine sediments might coupled with the past climate episodes where warmer, we normally have only one type of archaea, which is shallow water archaea, while cooler climates, we have a mixture of both signal from shallow and deep, which is quite an interesting finding, as we not normally use GDGTs to infer archaeoecology in the past. Normally, people do the phylogenomic trees to study evolutions of archaea based on their metagenomic data. But these techniques normally not reflect the dynamics of the archaeal abundances. So to be able to see the archaeoecology change over time and might corresponding or related to the climate episode, it's something that might giving us more insights on how the biogeochemical cycle changes in the past, as these marine ammonia oxidizing archaeas are key players in the modern environments. So that's pretty much the major implications from our papers. Seeing change of ecology through time, adding a new insight into the evolutions of marine AOA. And I think I'm gonna end my talk with these conclusion slides. So what we have done for the paper is that we have leveraged the use of GDGT data that normally people use them as a sea surface temperature proxy, but now we expand the applications. We use them to trace how these important archaeal group changes over geologic past. We have shown or established the link between archaeoecotypes and the archaeolipid distributions that suggested a coupled evolutions of ecology and archaeoecology and Earth's past climates. And that's probably what I have for you guys today. So if you're interested in read the full paper, here's the paper. I also have posted a summary thread of my paper on my Twitter, so you can follow me. The handle is paleolipidrr, and you have my full CV here as a QR code. And I would like to shout out for all of my co-authors in helping me publishing this first paper of my PhD study. I have learned a lot from these people. So Igor Zhang is my PhD advisor, and all of my co-authors, Dr. Ann Pearson from Harvard, Dr. Brian Hedlund from University of Nevada, Las Vegas, and Dr. Chuang Zhang from our Department of Oceanography, Texas A&M University. And with that, I would like to thank you, everyone in my lab groups and all the funding sources for my PhD studies. And thank you, PNAS, for waiving our publications fee and be able to publish our works to other people. Thank you so much.
Awesome. Thank you so much for such a great talk. So just a reminder to the audience that you can send in your questions via the chat to the questions at Paleoperks host. And we have one question in already. So I'll paste this into the chat so that you can see this. So our first question is from K. Sender Sawan, and Sender asks, how do GDGT ratios vary across hyperoxic and anoxic events, considering the effect of oxygen levels on predominantly anaerobic events? And can you comment on the effect of carbon dioxide degassing on such ratios?
That is an interesting question, and it's actually what I'm looking at it right now, because I also trying to understand how the oxygen affects the lipid distributions. I don't have a good answer for now, but I I still don't see something significant enough to say that the oxygen gonna shift, or how the oxygen concentrations affect our distribution patterns that we classified in our papers. I can talk a little bit that I try to plot if the person asking the question familiar with the paper from PNAS, Chin et al. 2015, where they have the incubation data with changing oxygen levels. On plotting that data on our 2:3 ratios versus text 86 value is showing shifting in text value, but not shifting in 2:3 ratios a lot. So it seems like oxygen impact text values, but not hugely impact 2:3 ratios. Not sure whether that answered the questions. And for the effects of CO2 degassings on the ratios, it's something that I have not looking into it right now, but it's something that I will be gladly discuss more for the person who asked the questions.
Nice, thank you so much. So the next question that we've had in is from Anya Hess. And Anya asks, she says, really cool work, Ronnie. My question is there any preservation differences between GDGT 2 and 3 that might contribute to the changes you see in the paleo records? For example, how quickly they degrade under oxygenated conditions?
These all of our interpretation is based on our current understanding that our GDGTs not shows a significant preferential degradations with the exposures to the oxic conditions from the paper discussed about it back. We have few papers, and I understand that the person who asked questions might familiar with these papers, but our interpretation is based on the fact that we believe that GDGTs are well preserved, not really have a significant preferential degradations between GDGT2 and GDGT3 in the ancient marine sediments. And before we plot all the data, we filtered out some of the samples that have abnormally ratios that we normally use, like BIT index or methane index or something like that, that we trying to make sure that what we see or what we are looking at is just the signal from the marine environments. But as for the questions, we assume that we don't have preferential degradations between the 2 to 3 different GDG compounds that we use for 2 to 3 ratios.
Cool, thank you so much. So we have another question. Which I'll paste into the chat. How do your records compare to other geochemical proxy records?
That's a good question. I haven't plotted our 2 to 3 ratios with other proxies yet, and I'm not quite sure if we have something comparable in terms of giving us the changes in archaeal ecology. But I am now thinking about the recently published paper talking about the delta N15 from planktonic forams. And that might be something that we can compare with, because the changes in delta N15 observed from planktonic forams suggest changes in maybe oxygen concentrations in the oceans, and that might giving us some clues whether changes in past ocean oxygen concentrations have some correlations with our 2:3 ratios patterns we see here or not. But I haven't really plot them together just yet.
It was good to have the next future plans. I'm sure it'll be interesting when you compare them as well. So there's another question in so another question from K. Sender Sarwan. Do GDGT ratios like the TEX86 indicator still hold good for younger time spans like the Neo-Archean and the Proterozoic as a proxy for temperature/water depth, especially due to the contribution of animals as predicted by molecular clocks and by sponge fossils interpreted by Elizabeth Turner?
I think for me, I might be biased because I study GDGTs. I would say we still able to use them as a temperature proxy. I think we probably know now that we're not going to get a great accuracy in terms of the absolute temperatures, which is something people in the community trying to improve our temperature calibrations and understand all of the uncertainties and stuff like that. But in terms of giving trends of warmings and rate of warmings, GDGTs still a good proxy. It's just a question whether we apply the reasonable temperature calibrations or not, when we now see that changing in archaeoecology might have some impact in terms of the distribution of GDGTs that we extracted from the marine sediments. For an example, our core top calibrations might good for a period of time when the archaeoecology is similar to modern day, while in the past, when the archaeoecology is quite different, we might have to find or improve our sea surface temperature calibration with TEX86 values differently. Or I'm not quite sure whether how huge of the impacts between applying one calibrations with the different calibrations, but that's something that we can do moving forward from this study.
Cool. So just see if any more questions come in. We've had a couple of requests for your PNAS paper.
I can send it to the steering committee and then distribute it, or whatever we are think the best way to do it, but I can share the copy of the paper.
Great. So we had one question in, which asks, which other sites would you like to explore to generate GDGT records?
I haven't had any maps here, but that is part of my other projects. As for people who study GDGTs, we know that we have a bunch of GDGTs over the past like 190 million years. But as you can see from this graph, there are gaps like all over the place, especially for the last 66 million years, even though we have a lot of data. But if we group them into like different latitudinal zones, we can clearly see that there are still gaps in the data from like low latitude regions or high latitude regions. And on the other ongoing projects that I have been working on, it's targeting those gaps in the Cenozoic records. So right now I have samples from one site from tropical Atlantic, and one site from tropical Western Pacific, and one site from like mid to high latitude and closer to the Tasman Sea. So that's kind of regional or like sites that I'm looking into right now in terms of filling in the gaps of the text 86 records, not particularly for these study, but it's something related to each other in terms of spanning the records or completing the text records.
Awesome. Thank you so much. So we have one last question, which is, where does GDGT lie on the spectrum of being a labile or refractory molecule? And can this position be used as a benchmark on determining the usage of other such molecules as paleoecology proxies?
I'm not quite clear whether the question is about labile and refractory in the modern marine system, like whether there's recycling in the modern until they're are deposited into the deep ocean, deep marine sediments or something like that. But from my understanding, I would answer that the GDGTs are refractorily marked molecules, which mean after they're deposited at seafloor, they're very stable. They're not degrade as easy as other shorter chain hydrocarbons. So that's the part of the reason why we found them in the sediments as old as like 190 million years. And it's with these refractory behavior, I think it's a good, it has a good potential to become a paleoecology proxy. It's still a lot to build on top of what we presented in this paper, but it's moving into a promising directions, if I would say. If that answers the question.
Nice. Maybe if the question asker stays for tea time, they can expand on more question. Great. So we haven't had any more questions in, so I will take over the screen share from you.
All right.
So thank you again to Ronnakrit for a really awesome talk. It's always great to see some organic geochemistry records within the series. And thank you everyone in the audience for joining us. And please remember to fill out the weekly feedback form. The link will be in the chat very shortly so that we can learn a bit more about who attended today's seminar, and join us next week on October 18th at the same time for a seminar by Onkuyu Nagash from George Washington University in the USA. And Onkuyu will be presenting a talk entitled Vegetation Structure and Modern African Systems, Implications for Homin and Paleo-Environmental Reconstruction. So just a quick reminder to fill in our participation feedback survey. The link will be in the chat shortly, or you can scan the QR code. And we'll also like to invite you to stay for tea time today. So this is just an informal conversation about the talk with our speaker in a relaxed setting. And our question of the week, which we'd like you to answer, is if you could time travel to any time period in geologic history, when would it be and why? So we're going to have a quick 2-minute break. So remember to get up, walk around, have a quick drink of water, come back in 2 minutes. And if you have paleo pets, we really like to meet them. So also remember to bring them to tea time too. We'll see you very shortly.